Thank you for the invitation to talk again, and I will watch that clock like a hawk. Because I'm a pathologist, I'm going to mainly deal with pictures. Apart from this, which is the college's definition of what pathology is, the college is what I pay hundreds of pounds a year to for no appreciable reason. So we're bridging between science and treatment. And really, I'm a histopathologist, which means I use tissue for diagnosis and to study the diseases in tissue. And I examine the tissue under the microscope, or cells, if you do cytology. And histopathologists, which is what I am, are responsible for making those tissue diagnoses or assessments that help clinicians manage patients from that tissue. I feel obliged to show some histology, so that is normal liver there. It's pink and purple, which I'll perhaps mention later. Oh, we've lost some of the heading, that doesn't matter. The important thing probably to take from this is that you won't ever knowingly meet anyone in the pathology department. But there's a hidden army, maybe 20, maybe 30 people that will be involved in the reporting, the processing of samples for any individual that come together to allow the report to come out, to allow the clinicians to actually action it. So pathology is hidden, pathology is physically hidden as well. So that's my front door. You can see there's no label on that, it's just all visitors report to reception. It doesn't tell you where reception is, it's actually behind you, so it's not even very helpful. So we're often on a mark, we're often in the basement, though fortunately I'm not. I'm going to follow, with pictures, the route that a sample, as it comes into the department, goes through before I end up authorising a report. So samples come into the department through the porters. They'll come in specimen pots, small pots for biopsies, big pots for something the surgeons provide us with. The pots are full of formalin or formaldehyde. That fixes the tissue. It basically makes the proteins go hard so we can do things to them more easily. The first thing when the specimen comes into the department is it's given a number. We have to track carefully every specimen so we always know who it belongs to. In Edinburgh we get 75,000 specimens a year, so tracking them is really, really critical. Once it's booked in, small samples get placed into little baskets called cassettes. Oops, they're about that big, and liquids can perfuse in and out of them. That's happening on the right-hand side. Big specimens, pathologists like me need to cut them up, examine them, measure them, describe them by dictating, and then sample the little bits that we want to see under the microscope. That's a cut-up bench on the left. It's got a downflow so that we don't bathe our lungs in formaldehyde. Once the little cassettes with the tissue in are fixed for another 24 hours usually, they have to go through processes. The machines on the left are our processors. The processors take about 4 to 5 hours to run with a run. It's a series of reactions that basically remove the water and the lipids that are in the cells and get it ready for subsequent processing. And that happens next on the right-hand side. So a processed specimen is taken out of a little cassette, out of a little basket, and then put on the top, and then flooded with paraffin, molten paraffin wax. That's a byproduct of the petrochemical industry, a useful byproduct, although you can actually use beeswax and other things should you really wish. Once it's cooled, that specimen in the paraffin wax is basically okay forever. If we can come back to a block that's 100 years old, cut it, looking at the microscope, and it will pretty much look the same. But we can't look at it without doing some other things to it first. So what we need is a very, very thin slice from the block of paraffin wax with the tissue, so that we can then, so thin we can shine light up through it in the microscope. And that's happening on the left-hand side. So these machines here are called microtomes. You mount the block in it, and you turn a handle very carefully, and it moves a very sharp blade up and down. It takes a very thin slice. So we typically section at 3 microns, which is 3/1000th of a millimetre. Those come off as a little ribbon, and I can't do it. It's far too difficult. It's like skilled scientists do this. Comes off as a ribbon, and then they're floated onto those water baths. And then you put a glass slide underneath, and then you lift up, and the section then sits on the glass slide. What we then need to do is turn it from basically something that's colourless into something that I can see under the microscope. So it goes through these auto stainers here, which give it pink and purple colour routinely. Those pink and purple stains have been used for over 100 years, some of them. One of them comes from tree bark. It just allows us to look at it and know what we're looking at. So eventually, the slides, nicely labelled, comes with a form that one of the clinicians, patient-facing clinicians, has filled out, appear in my tray, and I take them. There they are. There's some slides there. Take them and then look at them under the microscope. So that's my desk. That's my microscope. It's got a extended neck because otherwise I have to do that, and it's not very good. And really, the process of histopathology is looking at something and saying what you think. That's really all I do. We then have to dictate, or in my case, I just type. So we spend more time on a computer, and because I'm a liver pathologist, I actually spend probably more time looking at the patient record than I do at the slide, because I need the context. I need to know what else is going on. So I need to dig into the electronic patient record. So that's why I spend more time on the left of that than the right. Increasingly, laboratories are digitally scanning their slides, their glass slides. So pathologists are not using a microscope, they're using a computer to do everything. And that on the right-hand side is one of our slide scanners. That's for research purposes because we're not fully digitized, but we do that sometimes. Now, often looking at it on the pink and purple, the H&E is good enough. But sometimes we need to see whether there are specific proteins present that allow us to make a particular diagnosis. Sometimes that's useful to diagnose cholangiocarcinoma. Often it's best not to save tissue for molecular testing, but that's a different hobby horse of mine. If I do want to request special tests to identify proteins, they're done in the immunohistochemistry lab, so that's antibodies that bind to the specific proteins and go brown when we look at them. That's happening on the left there, and they use a series of autostainers to do this on the right-hand side. So there's lots of machines involved, and this is a particularly skilled and vital resource in the immunolab. Now, I mentioned molecular profiling. It's now routine, and unfortunately it's now routine in Scotland for cholangiocarcinoma. So that same block, paraffin wax with the tissue, can be cut again, and then the nucleic acid, so the DNA and the RNA extracted, which is what's happening on the left. In this case, they did pose for these photographs rather than me just taking them sort of stealthily. And then the extracted nucleic acid, DNA and RNA, is run through the machines on the right to generate sequences. So you're basically trying to read the sequences of the DNA and the RNA. And you get a lot of data from that. So you have to try and understand it. And that's very complex as well. So it then moves to a more computational assessment. And this is across the corridor from me. It's a good feature of the way we're set up in Scotland is the molecular lab is just across the corridor from us. So we work very closely with them. They use the sequences that come out of those machines and then integrate and understand and then produce the answer, basically. I'll point out, actually, Dr. Sophie McAfee, because she's been instrumental in setting up the molecular testing that we've done for cholangio. So we get that piece of information that comes into our report as well. It defines the abnormalities that are present, which should define the treatment that is available. They're basically weaknesses in the tumour that we can target with the treatment. Finally, and just as vitally, our administrative staff that never get any recognition whatsoever, but pulling everything together for MDT so that all the information is available in the right place at the right time when the treatment decisions need to be made. I was asked to discuss briefly the update in molecular testing in Scotland. So the last 2 years I've stood here and kind of shuffled and looked at my feet because we haven't been testing properly. We've done a couple of single gene tests. We had our proposal for testing approved in September 2023, and then we were told almost a day later, but there is no funding. So a conversation the following year here stimulated a whole sequence of action by MMF that Helen has mentioned in Holyrood. I guess it's your second home to Stansted. A couple of members' debates, an event last June, and then finally, in October, the health secretary stood up and said, yes, there would be molecular testing. So many patients have been involved and very helpful. Benjamin, I've worked with quite a lot, he's there. I'm going to mention the politicians because actually, in this case, they have been quite useful. That's Ben McPherson, who's one of Benjamin's MSPs. He's been very supportive. Marie McNair has hosted us at Holyrood a couple of times, and that's John Swinney, who's been re-elected as First Minister. All three of them actually been re-elected. We don't know what the composition of the new cabinet is, but we will be in contact with them again very shortly. This is the molecular testing that we're now doing in Scotland. I just want to mention briefly the organisation. So this isn't to scale. Shetland's miles further north. The ferry, we were talking about centralisation of care. The ferry from Aberdeen to Shetland takes 12 hours. Just the flight from Edinburgh to Shetland, to the south bit of Shetland, is the same time as the flight from here to Hamburg. And we're centralised for highlight surgery in Edinburgh, so that there are geographical complexities for Scotland. We're doing molecular testing in 3 centres. Edinburgh is providing it for basically this whole bottom chunk, including the west. Dundee are testing for Dundee and the Highlands, and then Aberdeen are testing for their local area and the islands. And what we're doing, if you think of DNA as a recipe in a recipe book, and the tumour cells is what, the dish that's being made, you don't take a recipe book to the supermarket, you write down your list and you go there. RNA is like the list in between. So some tumours have errors in the DNA, so tiny spelling mistakes in the recipe book. If we read the recipe book by DNA sequencing, we can identify the errors and see which vulnerabilities can be treated. Some tumours have even more disrupted recipes. So if you remember an episode of Friends where Rachel's recipe book has 2 pages glued together. So she makes a combination of a trifle and a shepherd's pie. That happens in these tumours, where we call it a fusion, where 2 bits get joined together. Now, we don't read the recipe, but we read the actual shopping list, which is the RNA sequencing, so we can identify different targets there that are vulnerable to particular drugs. And then sometimes we just test the actual protein, so test the actual tumour cells. So HER2 is an example of that, by that immunohistochemistry that I mentioned. And that's my second histology picture. This is HER2 staining. So patients where the tumour is this brown everywhere from the membrane are the ones that are going to benefit from the anti-HER2 agents. I'm just going to kind of finish with the research side of it. So I believe that most of the information we need to know about a tumour is present in that tissue. So after I've done diagnosis and we've done the sequencing, we've got the treatment information, those blocks go off and are stored for up to 30 years. They're actually mostly, for most of the UK, stored in a climate-controlled storage facility, which is in an old mine in Wales. There's no use to anybody, that tissue there. There's information in there. It's our job to get it out. So our bioresource and the bioresource network in Scotland is a way to access that information, access that tissue, and present it and make it available to researchers to use. And I know it's not just us saying we should do that. I know that the patients think this as well, because we, in the last 2 years, led by Rachel Guest, we are prospectively sampling, our kind surgeons are prospectively sampling resections, lesional and non-lesional, when they do them. So they're consenting the patients. And I think we've had 199 out of just over 200 patients. So that at the lowest point in their lives, they have said yes. They want to help. They're not helping themselves. They want to help others. And so this is the resource that we've collected, and I've highlighted cholangiocarcinoma in the number of cases that we've sampled. So we don't always have lesional tissue. And this just reflects our resections. We're not actually sampling from explants at the moment. I've got a couple more slides, but I'm going to stop at that point because I think that's a nice place to stop. So pathology should be the answer if we can extract that information. So thank you.